by thanking the organizers for the opportunity to share my work with you on SABRE, hyperpolarized pyruvate, where we re-examined the binding behavior of pyruvate by analyzing its exchange dynamics. So, wait, okay, cool. So everyone, I think, knows what means SABRE, signal amplification by reversible exchange. So it's an NMR hyperpolarization method. It works by bringing together a parahydrogen and a substrate into a iridium complex where they form a J coupling. Here's the twist. The substrate doesn't react. It binds, gets polarized, and leaves unchanged. This means one thing, continuous hyperpolarization. Recently, SABRE have a new ground with pyruvate, achieving signal boost around 20%. But if we want more, and we do, we need to dig deeper. And the key is understanding and measuring J couplings and exchange dynamics to unlock even higher hyperpolarization yields. So we started first by developing a toolkit, a clear methodology to quantify exchange rate within SABRE systems. So to do this, we started with like the normal 1D, 2D NMR spectroscopy techniques like noisy and XZ. And our main target was to transfer polarization to the bound substrate. And with a demix, like mixing time or evolution time, we can follow how polarization are exchanged with free form. Then we will fit these data with the block mechanical equations to extract the exchange rates or dissociation rates. So basically, we started with a known substrate like pyridine, because these substrates or nicotinamide, because their exchange rates has already been reported in the literature, and basically it will allow us to validate the methodology and make sure that it provide accurate and consistent results before applying it to pyruvate. And so the variation of time or evolution time and using the block mechanical equation showed a Repuse method to quantify chemical exchange. However, on the other hand, as we can see, so basically we are using thermal measurements, acquisitions, and the chemical shifts difference between the bound and the free substrate are very small. So the chemical shift dispersion is very small. So it caused us some problem for acetonitriol or metronidazole, not every substrate, to measure the dissociation rate, because it was difficult to clearly integrate the free and the bound form. So in this case, it's easy to move to hyperpolarization. So we made sure here to we compare two hyperpolarization pulse sequence like PHI-NET plus and esoteric. In these two pulse sequence, we know that it will create a net magnetization on the bound substrate. And then with the methodology of having an evolution time, we can study how a polarization will be transferred or exchanged between the bound and the free form here. And for sure, we had we measured for almost like also acetonitriol or metronidazole substrate, and the chemical shift dispersion was very wide, like 50 ppm, and sensitivity was even very high, that we were able to measure pyridine at natural abundance isotopic. And for sure, by changing the temperature, we were able to calculate the activation enthalpies and entropies. So now we have the methodology. We move to pyruvate SABRE. So as we know, after activation of pyruvate SABRE, we have three major complexes. Complex number one, where we have pyruvate binding on the equatorial side. Complex number two, where it binds on equatorial and axial sides. And complex number three, it doesn't have pyruvate, but it has 2D-MSO instead. And after Pasadena, we can clearly see 6 antiphase peaks, which proves the existence of these three major complexes. And these assignments represent the structure which previously proposed in the literature, which will be revised by our experimental data now. So we start with 1H thermal measurement. So we change temperature, and we see when the temperature was increased, complex number 1 was dominating in the sample, which make it thermodynamically less stable comparing to complex number 2 and complex number 3. Also, we we increased pyruvate concentration in the sample, and we see one is increasingly, it was like dominating, and which contraries the model, because if we consider that also complex 1 and 2 has a bind pyruvate, should be at somehow increasing both together. And when we applied increasing the MSO concentration, we found like complex number 2 and 3 are dominating, and complex number 1 is decreasing very much. So this behavior contradicts a little bit the structure before assigned for complex number 2. So in this case, we were motivated to study more or develop a pulse sequence to study this exchange that's happening within the complexes. So our pulse sequence were developed. So after Pasadena, we have both hydrides polarized. Then if we apply the first part SEP, we make sure that we transfer polarization to only one of the selective hydrides. And after SEP spin-up, we create an antiphase between the selective hydrides and the carbon. And with this, we were able to measure 6 J coupling less than 1 Hertz, which were before experimentally not done, only was guessing by DFT. And then with the last part, we apply 180-degree, we have a net magnetization on the bound pyruvate, and then with the evolution time, we study the exchange. But here, the surprise is that the bound pyruvate doesn't go dissociate completely to free form, but it's changed binding to complex number 2. You can see, so it switched binding, then become free. So we introduce a general scheme for pyruvate SABRE to provide a basic picture on the evolved process. And here we showed a hypothesized reaction pathway that allows the equilibration of 3 complexes. So first we change the numbering of complexes. So now complex number 1 is the one which has 2D MSO. And here we showed a series of dissociation reactions of evolving of loss of chloride, and then we have intermolecular hydrogen exchange between 1 and 2, and also between 2 and 3, and also we have the MSO exchange between complexes and pyruvate exchange. And here we have a more simplified version. So it includes the major 3 complexes, but now complex number 2, the one we considered, is a monodentate pyruvate binding instead of the previous estimated. So now here we call it complex number 4, but in the literature it was complex number 2, where it was a bidentate pyruvate binding. But in our experiment show it is absent and energetically unfavorable. So we will focus on our 3 major complexes, 1, 2, 3. And number 3 is what previously was number 1, which has a pyruvate binding on the equatorial side. So as we said, after applying SEP spin-up plus SABRE, we fit using the block mechanical equations. And as we saw with the MSO, so we noticed that while increasing the MSO concentration of the sample, we saw complex number 3 tend to switch binding more faster to complex number 2 before its complete dissociation. And at high DMSO concentration, we have more stabilizing of complex number 2. And we also did a SABRE, so we studied the exchange of hydrides, so to see whether also we can see this effect. So if we hyperpolarize one of the hydrides, and with the evolution time, we were noticing, for example, this example of complex number 3, so it switched to other hydrides in the same complex, then to hydrides of the complex number 2 before it become completely dissociate with hydrogen H2. And also we can find here the same exchange rates. So from complex number 2 to complex number 3. And this is the orange here, for example, between both hydrogen of the same complex, and purple is a complete dissociation. And this is a scheme just to see that HA, or for example, HA was exchanging with HB, and both of them was HA and HB from the second complex, and then with the free form. And also to prove that complex number 2 has indeed 2D MSO instead of 1. So we have the pulse sequence to study the SAB-spinap, like to measure J couplings. So we apply it to we use 13C labeled the MSO, and we measured interaction between hydrides, and we found like there's 4 interactions with one of the carbons, and from the second hydrides we only have 2. And also it's not only like we were able to measure for one delay, but we can also follow the evolution of all 4 J couplings. And this is another proof that complex number 2 has indeed 2 DMSO instead of 1. So with this, we already spoke about the conclusion. We were able to measure J couplings for the first time. Instead of guessing with the FT, we measured J couplings between the hydrides and the carbon of the DMSO and complex number 2. We had a evaluation of pyruvate binding and exchange in SABRE, and we had a refined structure of complex number 2. With this, I would like to thank all my contributors, and also thank you everyone for your attention.